Hi, my project this summer was on examining the characteristics of the erecta mutant phenotype in Physcomitrella patens. Our lab mainly studies how organ size is regulated. We want to know what pathways contribute to development, and it's important to know this especially when we're considering crops. So we mainly study mutants that influence cell growth, and in this example we'll be examining the phenotype. So Physcomitrella patens is also known as the spreading earth moss. It is very useful for examining plant growth and development. It's very easy to grow in the lab because we put it in a petri dish and you don't have to do much to care for it. Another advantage is that the moss has a lot in common with vascular plants. So again, it's easier to care for, so it's very nice to use in comparison. And the two-dimensional nature of the leaves is very helpful because when we're using our imaging, we only have to care about one layer of the cells. So my mentor, Joe, attempted an erecta-like sextuple mutant with CRISPR-Cas9. So he mutated 6 erecta-like genes and created this mutant. Erecta is a leucine-rich repeat receptor-like kinase, which we will go over in the next few slides. So, as you can see in this image, the mutant is much shorter and has very round leaves that stick like straight up, whereas in wild type they're very long and they branch out, which we will consider. Right now we want to know why that's happening, but another mutation could be causing this phenotype. Because when we mutated all of these genes, they were close to other genes that could have also been altered and caused this. So most of what we know about erecta is from studies based on Arabidopsis. In Arabidopsis, there's a very similar phenotype. They grow straight up, and they're much shorter. It influences photosynthetic capacity, cell elongation, asymmetric cell division, and flowering time. And in Arabidopsis is most comparable to our mutant by the triple mutant, which you can see on the right here. It's much shorter than the single mutant, and you can see it enlarged over here. So in this study, they took a look at downstream signaling events caused by the erecta receptor-like kinases, and they looked at the pedicels, where we knew that erecta causes a shorter pedicel, but we wanted to know what the cells were doing in that case. So we noticed they had a smaller cell number and they had bigger cells, which was them trying to compensate for having less cells, but it was still shorter overall. So the aim of this project was to analyze the effects of the erecta mutant and get a better understanding of the phenotype. Our hypothesis was that the phenotype was caused by shorter cells and a decrease in the cell count. And this would suggest that the erecta mutant limits cell growth and challenges the cell cycle. So to examine these characteristics, we used a laser scanning confocal microscope. The leaves of the wild type were imaged in order, and the shoot of the artichoke was imaged all at once. And we did this because the wild type is very large, and because the leaves bend, the best way to look at it under the microscope was 1 by 1. And for the artichoke, because the leaves are more fragile and shorter, the best way to image all the leaves was all together. The downside of this, however, was that we weren't able to get a cell count of all the leaves of the artichoke because the other leaves would cover it up. And we also used PI to do our imaging. So we use morphographics to do our analysis because it allows accurate cell measurements with curved surfaces. We would first brighten it, and then we would use edge detect, which would create a 3D shoot for us. Then we would select which area we wanted to do. So in the case of the erecta, when we're using the shoot, we would find one leaf, because we wanted to do it 1 by 1, and generate the curved surface. And then we would project the cells from the original shoot onto the surface. And the next step would be to seed the cells. So we would seed the cells by drawing onto the leaf itself, and then morphographics would register, and we would run the segmentation function like you see here. And then our next step would be to use the heat map, and the heat map would gather information on the area, the aspect ratio, and any other specifications that we wanted. Right off the bat while we were imaging, we noticed abnormalities with the artichoke mutant. As you can see in wild type, the midrib goes through most of the plant and is very straight and linear. But in the erecta mutant, it goes about halfway and it splits off into two. And this only happens in some of the erecta mutants, but it's not something we noticed in wild type. Another thing we noticed is that while in wild type, the leaves or the cells will grow in this direction along with the rest of the cell, or along with the rest of the leaf. We noticed that in erecta, while some of it would grow like that, the other parts would go in perpendicular, which would lead to this distortion of the cell border. We also noticed that there was much more frequent branching in the erecta shoots. So in wild type, this would also happen occasionally, but not nearly as frequently. But we would see other shoots start growing out of the base of the erecta shoot. So in terms of area, we noticed that the median wild type was almost 300 square microns larger than the erecta. And while both of these histograms are skewed, you can still see this difference. For the erecta median, it was 1,008 square microns, and the wild type median, it was 1,278 square microns. And this supports our hypothesis that the erecta leaves would be shorter and smaller than the wild type, and it also could explain why the entire shoot is smaller. Our measure of shape was the aspect ratio, which is just the length divided by width. As you can see here, it's typically going to be the larger one divided by the smaller one, and it gives us a sense of long versus round. So the rounder ones and the ones that are more circular and square shaped will have a aspect ratio of 1, and that'll be the most compact. So we noticed that the erecta mutants had a much smaller aspect ratio, meaning they're more round, and that the wild type were much longer. And this suggests that the erecta cells have fundamental growth differences, and that the rounder cells are leading to the rounder shape of the artichoke mutants. So in conclusion, the erecta has much more branching, margin defects, and midrib splitting than the wild type. It also has a smaller cellular area and contributes to the smaller size of the aspect ratio that indicate these cells are much rounder. These suggest that the erecta-like genes, gene mutations are causing a severe growth and developmental issues. And once we are certain that the erecta-like gene mutation causes this phenotype, we can begin to go further and see how it works in other plants as well. So we are interested in using live imaging, especially to compare the developmental differences between wild type and the erecta. We also want to see how the midrib splitting occurs and how the growth differences occur. We also need additional studies to confirm that the erecta-like gene mutations are what is causing this phenotype. And we would need a further analysis of other factors that could contribute to the shoot and leaf size, like how we weren't able to measure the cell number because of the issues. So I would like to thank my mentor, Joe, and my principal investigator, Adrian Roeder. And I'd also like to thank NSF for providing grants to fund my research, and UTI and Cornell for having me here for the summer. Yeah. Mm-hmm. I think they were pretty similar. We didn't really study them. Okay.
Do you only see a phenotype with all 6 mutations, or are there intermediate effects?
We haven't looked at the other effects, I don't think.
You were straight through all 6.
Yeah.